And the final flash talk of this session is going to be given by Adam Reyment from the University of Cambridge, who's going to talk to us about determining the role of s2m in the replication and pathogenicity of avian coronavirus, and Pirbright as well.
Yep. Hi, everyone. So as mentioned, my name's Adam. I'm a second-year PhD student in the coronavirus group at the Pirbright Institute. And today I'm just going to give a little bit of an introduction as to what I've been doing in my PhD project, where I have been trying to determine the role of the s2m in the pathogenicity and replication of infectious bronchitis virus. So what is the s2m? So the s2m is an RNA structure that's found in the 3' UTR of many positive sense RNA viruses belonging to the Coronaviridae, Astroviridae, Picornaviridae, and Cliciviridae. And as you can see by this alignment here, representing many viruses from these families that harbour an s2m, the sequence is very well conserved between these viruses and therefore may have an important role in various viral processes. So as mentioned, I focus on IBV, which has a lot of benefits, including the fact that we have multiple systems in order to characterize the mutants that we generate when investigating the s2m. So, for example, we have many primary and continuous cell culture systems set up. We have a very well-established ex vivo tracheal organ culture system. We can look at some growth kinetics in ovo, and then very importantly, we have the ability to look at the virus in the endemic host through animal studies. So my project focuses on 3 strains of IBV. So Bidet, which is a heavily lab attenuated strain that doesn't cause clinical disease, M41 and D3A, which are both pathogenic to varying degrees and are also endemic in the UK. And to try and investigate what role the s2m might have in the viral process of IBV, we generated a range of mutants, and I'm just going to talk about 2 of them today. So the first one is a deletion of this highly conserved GCC trinucleotide motif here, and all 3 strains of virus with this mutation were able to be rescued and characterized. And then the other obvious one was to delete the 41 nucleotides that correspond to the s2m sequence. And so far, through many rescue attempts for all 3 strains, this has not been successful. But going back to the deleted GCC mutant, so again, as I mentioned, we have the ability to characterize these in vivo. And what we see is slightly different between M41 and D3A. So on the top here, we have the clinical presentation and cellular activity for M41, and we have the deleted GCC virus in red and the parent in blue. And as you can see, deleting that trinucleotide motif appears to have a significant effect on clinical presentation over the course of our animal study. So as you can see, the snicking is heavily reduced, and there's a complete abolishment of rails, which is a rattling of the bronchi. And then also on top of this, when we look at the cellular activity of trachea isolated from the chickens in this experiment, we see a move towards attenuation in terms of the cellular activity. For D3A, this is not the case. So it's a little bit harder to see on here. But for the deleted GCC virus, in terms of the clinical presentation, it behaved very similarly to the parent virus, both in terms of the snicking and the rails. And as you can see by the cellular activity, throughout these 3 days, so days 4, 6, and 10 post-infection, they had cellular activity levels very similar to the parent, indicating that this is not an attenuated virus. So if you want to hear a little bit more about the s2m and its potential role in the viral processes of infectious bronchitis virus, please come to my poster. It's number 12. And a big thank you to everybody in the coronavirus group at Pirbright, also my lab in Cambridge, the Lyra lab, and thank you to the UK-ICN for inviting me here to share my research. Thank you.